Okay, so I'm going to talk about the study of Rett syndrome in terms of synaptic metabolism and how we are trying to search for new therapeutic options based on this concept. So first of all, we're talking what's synaptic metabolism. Okay, so synapses, we all know, is like the way of communication one neuron to the other. It's a process, like synapse is a process by which two neurons communicate, and what we define like synapses are really complex processes um, in which multiple reactions take place, including all the basically recycle systems, all the receptors docking and production, all the energy needed for the synapse to work out. So all those biochemical reactions that make the synapse to be a synapse are what we call synaptic metabolism. And we are trying to study diseases from that perspective, like trying to study what's going on with the synaptic metabolism with all those biochemical reactions to understand the disease instead of going to the symptoms to the disease. And based on that, we also are studying, among those diseases, Rett syndrome. What's Rett syndrome? Rett syndrome is a neurodevelopmental disease in which many events take place, including seizures, loss of speech, and purposeful use of hands, apnea, stereotypes. It's mainly a neurological disease. This is children, mostly females, are born normal, are born like phenotypically normal. They have a normal early development, and at the age of 6 months, they start experiencing a regression on their development and experience all those symptoms. And what's happening? Like, why is it mainly neurological disease? This is because most of the cases are due to a mutation in MYC-P2. MYC-P2 is a X-located gene, and it's in the X chromosome. And it functions as a master regulator. It binds to other genes, and sometimes it inhibits the expression, and in some other genes, it enhances its expression. MYC-P2 happens to be involved in many pathways, both regulatory, of expression, the acetylation. Is like those are some of the pathways that it's involved in. So when you touch MYC-P2, you touch everything in the cell. And this is what's happening. Like, to approach the disease, we need to break it down. It's such a complex disease, you need to break it down into events to try to study that. So pretty much, we could divide the neurological dysfunction in this case in 4 terms. First of all, there's an imbalance in the neurotransmission. There are some neuroinflammation processes. There's a defect on the bioenergetics. And there's a problem with neural maturation, among many others, but those could be like 4 pillars of the disease to target. So starting for the first one, we're trying to address all of them to treat the disease or to find new treatment options. So regarding the neurotransmission imbalance, all the systems, all the neurotransmitter systems are unbalanced in these kids. They have everything to be done, but the main attention should be put towards the glutamate and GABA neurotransmission, as has been shown to be responsible for the main characteristic of the disease. So regarding that, well, glutamate is the activator, the synaptic activator. GABA would be the silencer, the silencing neurotransmitter. So they will, in a fine tune, they will both equilibrate the communication, saying go on, go off. And in this case, it's unbalanced in terms that GABAergic neurotransmission is not properly working. So and glutamate neurotransmission is enhanced. So they are like hyper enhanced. So regarding GABAergic neurotransmission, GABAergic neurotransmission is quite a complex system, as all of them, and we started to put our attention towards the ionotropic receptors. Well, we know just going fast that receptors can be metabotropic or ionotropic. Those account for the slow neurotransmission, those for the fast hyperpolarized neurotransmission. And GABAergic ionotropic receptors are a huge family of receptors with many subunits forming pentameric receptors. Those are the target for many known drugs. And why we did focus on those? Well, they inhibit the action potential. And why did we focus on that? Well, because GABAergic neurotransmission, to be an inhibiting neurotransmission, it depends on the chloride concentration on the neuron. And it has been known that during development, there are 2 channels, KCC1 and KCC2, that switch the production, the expression, to make GABAergic neurotransmission from positive activator to inhibitor. And it had been shown by our group quite a few years ago that this switch from activate neurotransmission to inhibit neurotransmission is not happening in MECP2 patients. So we started to accumulate evidence that there was something that we could do there. And also because all the scientific community is pointing towards this neurotransmission as a big pillar of the disease. Meaning, well, it was described how the GABA dysfunction signaling mediated some stereotypes in Rett syndrome phenotypes. And also more important, how MECP2 restoration, expression restoration in GABRAIC neurons was sufficient to rescue multiple of deficiencies in mouse models of Rett syndrome. This is really important because this says that GABRAIC is important, and also if you restore GABRAIC neurotransmission, you can target the disease. So that's pretty big. Well, also there were like many evidence saying like how GABRAIC neurotransmission was affected, but yet all of them, like many of the conclusions were like it's somehow affected, but we don't really know how and when it's affected. So our two main questions at this point were, well, how is MECP2 function affecting the GABRAIC receptors themselves? And second, can we actually, based on that, define a new therapeutic target? So we did set for this in collaboration with the EDVL and Neurodevelopment Lab at the University of Barcelona. We did set a model of 4 different scenarios of growing complexity. This is, we started on simple cells, then we moved to neurons, to the animal models, and to brain samples. So starting single cells, what we did was a really simple experiment in which N2A cells, which are like transformed immortalized culture cells, we first of all inhibited or blocked the expression of the endogenous MECP2, and then we re-express either MECP2 in the human form wild type or mutated. So first of all, we did see that when we inhibited MECP2 expression, we did see a decrease in the expression, and when we re-expressed the human wild type form, we did see an increase in the wild type, which will not happen when we express the mutant form. So this pretty much said, okay, we have a valid system of inhibiting and re-expression. So with this system, let's see what's happening in the receptors. We took one subunit, GABA1 receptor, which is the main subunit of these ionotropic receptors, and we did see that when we overexpress the wild-type form of MECP2, we accounted for a huge expression of this subunit. This is, MECP2 is actually working as a positive regulator of GABRA1, which is this GABA receptor subunit. Then we next switch to a more complex model. This is culture neuron, primary neurons. Well, they are not well seen because of the light, but what we did is the same. Let's inhibit the expression and re-express in the wild type or mutant form. And we did see, first of all, that we could do that, like re-express MECP2. And that when we did that and targeted GABRA1, this is GABRA in green and in red and GFP in green, only when transfected with the wild type form, we will see the re-expression. This is, when we express a mutant form of MECP2 in a culture neuron, we didn't get to express this subunit, this receptor subunit. This is, again, MECP2 works as a positive regulator of GABA1 subunit. Yeah, this is all the same, but just to be published. So with this, we concluded that there is a straightforward relationship between MECP2 and GABA1 receptor. But what happens here is that we were working in a very, very simple model. And we have to take account that Rett syndrome is a neurodevelopmental disease. This is, kids are born normal. When they are 6 months age, they start doing the regression. So we cannot focus on just culture models. To get that, what we did was work with the mice to see how was this affecting during evolution, and with that, like, increase the translational value of the study. We work with female mice at 2 times. P33, this is 33 1-month-old mice, which are female mice, which are asymptomatic, and 6 months mice, which are already symptomatic. And we targeted MECP2 expression and GABA1 receptor. And we did see a decrease on GABA1 receptor expression at 1 month, which was not accounted when the mice were symptomatic. This is really important because so far studies have been done with the symptomatic mice, which is what you want to treat. And what we are seeing here is that some of the main alterations are actually in pre-symptomatic individuals. and also that these results, the results we are observing, are temporally dynamic. So at this point, we know there is a straightforward relationship between MICP2 and GABA receptor. Then we have dynamic results and pre-symptomatic damage. We say, okay, let's see how is the GABAergic pathway in the patients. As for that, we perform a few experiments of RNA-seq. This is, you take all the RNA in a sample, put it to cDNA, and measure the expression of every gene you want to measure. We did that with the brain of 2 patients who had died at 15 years old at the hospital. So we had the brain sample at the hospital, and we analyzed these brains. The only problem, one of the problems, we didn't have a control of a healthy brain, because you don't have healthy brains. So what we did was compare the results first with an inter-experiment with another patient for an experiment control, and then compare the results to all the publicly available results. So this is, you cannot say with that like how many times it's incremented or decreases, because well, you don't have that such exact data, but you can say the tendency, how fashion it's at. And what we saw, it was in those patients a tendency for overexpression of the result. At first we were surprised for that, because we had seen first a decreased expression. But when we thought of that, well, we analyzed the rest of the pathway, like the whole pathway, and overall, we actually saw an or non-modified or overexpression of most GABAergic genes. So we were a bit surprised by that, but when we thought of that, what we had seen was temporally dynamic disease. We were seeing patients at the end of the disease, like who had already died. So yeah, that's not that surprising if you think of that. Like it corroborates our results that when you are pre-symptomatic, you might have something we had seen in mature models, but when you actually grow through the disease, you get another symptoms or another phenotypical symptoms. So pretty much, MICP2 acts as a regulator of GABA1 receptor. There is a huge importance of the time frame, and GABA1 could be a therapeutical target with deep need of putting the attention towards before the disease, like early diagnosis. Well, this is to be published. This is under revision now, and actually this study has been funded by Mi Princesa Rett, which is a patient association. So it actually increases like the value for us, because it's for these patients straightforward. So we say that there was this neurotransmission imbalance, and also neuroinflammation, bioenergetics, and neuronal maturation. So regarding all the other ones, we're actually working with minorics and in collaboration with some other groups too, with MIN 102, which actually just has been exposed before. So I'm not going through how this works, because we have already been told. We'll know it's PPR gamma agonist. And what we want to do here is to counterpart the oxidative stress, increase the mitochondrial biogenesis, and decrease the inflammation. So well, first of that, to know that we know through the literature that this might be affected in Rett syndrome, but we need first to set the ground base to our disease or to our models. So again, we have set up a scenario with increasing complexity models. First of all, in cells, this time it would be fibroblasts of the patients. Then we move towards neurons of the patients. This is through iPS cells differentiated into neurons. And finally, to the mice model of Rett. And we are first going to look for the mitochondria, and then for the neuroinflammation and whole brain affectation. So regarding the mitochondria, we have set 5 parameters, which will be the oxygen consumption of the mitochondria, the ATP production, the reactive oxygen species generation, visually shape the shape of the mitochondria through electron microscopy, and the dynamics of the mitochondrial network through different dynamical markers. So regarding the respiration, we have done a few experiments of a technology called CHOS, which is like plate respirometry. You measure the oxygen consumption in response to different drugs, namely oligocin, FCCP, and antimycin A-aminotenone. And yet these are preliminary results, but we did see a decrease in the oxygen consumption. As for these are extremely preliminary, so shouldn't be taken into huge consideration. But we do see definitely some alteration at the oxygen consumption. Yet real preliminary it is. And these go together with the ATP consumption. This is, okay, your mitochondria are consuming less oxygen, but what's happening with the ATP, which is like the energetic function. And for this, we actually can rely on this. We did see a huge decrease up to almost half, 50%, on the mitochondrial ATP between Rett patients and controls, but Rett fibroblasts and controls. This is, we do have fibroblasts, or due to MECP2 mutations, we do have a decrease in the ATP production in these cells, like great decrease. This is good as, well, this is bad, but this is good in terms that we can target that to try to solve something. We next move to the reactive oxygen production. We studied first the anion superoxide anion with the probe mitosocs, and we saw an increase of like almost two-fold increase of ROS in the patients compared to the controls. When we analyze the detoxifying enzymes, this is superoxidase mutase and glutathione peroxidase, which would take care of the reactive oxygen species and release them from the cell. We saw no reliable differences between those two enzymes and patients and controls. And then this, quite a shame. Can I puedo apagar la luz ahí, quizá? So in controls, we did see, well, it's not really well seen, but you have to trust me. We did see the shape in tubular mitochondria, like, you know, the typical mitochondrial network you associate with healthy control, while in Rett patients, we saw mitochondria in donut-shaped mitochondria, which are usually associated to stress and energetic deficits and poor docking on the receptors. So it did fit in terms that yet we have to consider that these are fibroblasts and not the actual neurons. But if in the fibroblasts you are seeing these, and fibroblasts are not a type of cell that relies on mitochondria much, what's going to be happening in neurons? If it's the same, donut-shaped mitochondria are associated with poor docking of the receptors and inactive synapses. So it makes sense. It actually fits with the expected results. We analyzed some markers associated with fusion and fission events. As for fission, we had DRP1 and FIS1, and we did see a decrease in the expression. Well, not for this patient, but for these two, and in FIS1. Decrease in fusion, and we studied mitofuscin 2, which is a fusion marker, and we see an increase in fusion. So, yeah, sorry. Pretty much summing up this part, we did see a decrease in ATP synthesis, a decrease in oxygen consumption, an increase in superoxide and in ROS production, and an altered dynamic in donut-shaped mitochondria and increased fission. So next would be analyzing all these events in the treatment with MIN 102. When we have that, we will move toward more complex systems, which will be iPS cells, and then to the mice models. Well, I'm the one who have explained this, but that's not me who has been doing that. Well, I have doing that, but I'm part of a big team in which we have like clinical people with Mar O'Callaghan and Dr. O'Callaghan here and Merce Pineda also. We have the genetic team with Dr. Judith Armstrong. We have neuropsychology team also, and the molecular lab. We have a huge team, and we are led by Dr. Ángel García Cazorla. Like, the brain behind all this is that. And we are funded by, well, mainly funded by different agencies, and also usually collaborate with several groups. So that's it.
And this is it. Thank you very much.